because I forgot the first day. Even though I had it, I still didn't do it. Oops. Does anybody have any questions before we get started? We're going to jump into the PowerPoint from Monday. Haley, you doing okay? Okay. Okay, well then let me share screen. Okay, try to get where I can see everybody. Okay. So we had started talking about plastids. I think that we covered this slide, but I'm going to start here again to kind of bring us back into where we were. So plastids, probably not something that you think about much because in humans, well, in animals, we just don't do plastids. There are a number of plastids. The one that you're most familiar with is going to be the chloroplast, and it's the most conspicuous. It's actually rather large. We've seen that in some of our diagrams. It's larger than the mitochondria. It's not larger than the nucleus usually, but it's quite easy to see under the microscope. Sorry for my yawning. Apologize. Can anybody tell me about the double membrane? We mentioned this last time. Why do we have a double membrane? It's evidence of a word that starts with E. Somebody other than Elena, because she gave me the answer last time.
Endosymbiosis.
Yeah, endosymbiosis. And can anybody remember what organism may have been the original endosymbiont absorbed by the plant cell? Starts with the C, ends with the bacteria. There's only 4 other letters I'm missing. Nobody? Cyanobacteria. So cyanobacteria, what's another name for cyanobacteria? Does anybody know that? There's kind of a common name that we use sometimes. So, and it's basically based off the name of cyano. If you saw a crayon that said cyano on it, what color do you think that would be?
Blue.
Blue. So we call these the blue-green algae, cyanobacteria. Some people call it blue-green algae, which is not technically true. They're not alga. They are actually a bacterium. So if you do hear blue-green algae, that's not quite right. But a lot of times you see them growing in the same places, like on the scum of ponds. You'll get those together with other algae. Okay, now I mentioned the colors because we know colors make a difference in photosynthesis, right? We're going to get into that. What color is the chloroplast? It's green. Now, inside of that double membrane, there is an area called the stroma. There's a bunch of enzymes filled inside there. There's DNA, RNA, and ribosomes, which is also other evidence for endosymbiosis because you've got DNA somewhere other than the nucleus. That means these probably were things that were living at other points separate from this plant, or in this case, maybe an algae or something. There is also something called grana, which are made up of thylakoids. The grana are the stacks of thylakoids, and these are membranes that actually have the chlorophyll molecule embedded within them, and they have very large surface area. Okay, their whole purpose, which we'll get into more later, is photosynthesis. So we're just looking at inside the cell. We'll come back to the specifics of photosynthesis later, but know that chloroplasts are really important. You can see the green ones inside of this. I think this is, yeah, it's a spirogyra. Okay, that's an alga. Okay, so chloroplasts can move within the cell. They actually have a framework in which they can move along and move things around. So in dim light, notice the way that they're oriented within the cell. Look in bright light, look at the way they're oriented. Any ideas why the chloroplasts would orient this way in bright light and this way over here in dim light? No ideas? Well, do chloroplasts absorb light? Come on, I know y'all know the answer to this one. Do chloroplasts absorb light? Tyler's like saying, yes, they do. Good job. Yes, they definitely do. Okay, because what's the purpose of photosynthesis? To absorb light, right? And then to make food. So why move out of the way for light if it's really bright? Well, you can have too much light. They can actually be damaged. And so they have the ability to move around in the cell to control the amount of light they absorb. If it's in dim light, they'll place themselves in areas of the cell to try to absorb as much light as possible. And in bright light, they'll try to move their cells in such a way that they don't absorb too much light. Okay. You can see the stacks of grana here. Notice they're all connected in one big membrane. They just get stacked on top of each other. That is to increase surface area. The space in between, so all this white space in and around the thylakoid is actually the stroma. So the space in and around, you can see the double membrane, but all that space in and around, that's the stroma. A couple of other plastids, the chromoplasts and the leucoplasts, are also additional plastids. We won't get into a whole lot. We will look at some chromoplasts in lab. We were going to do that this past week, but I think we were going to run out of time. What do you think chromo refers to? What's that, Tyler? I saw something. You want to share? No? Okay. If you see something that has chromo in the word, what do you think that prefix basically represents? And I'll let you think on that while I say something else. This is really important, I think, especially in botany, but I think in all of science, true and too. Um, when you see things like chromoplasts, thinking about the prefix and the base word actually tells you a lot about those words. So you're breaking those words down into, yeah, Kibiana, that's right, exactly, it's color. Um, so color, and we know what a plastid is, right? That's what plast represents. So is this a colored plastid? That's basically what the name says, right? So we've got chromoplast. So in science, and I think especially in botany, being able to break the words down tell you a whole lot, especially the names of the plants. Um, so learning, and a lot of this you already know, you just don't think about it, but learning these prefixes, suffixes, um, and trying to pay attention to the root words, you'll get a lot of information. Sometimes you'll see a brand new word that you've not seen before, but you'll be able to figure it out. Um, does that make sense for everybody? Okay. And have you ever wondered why we make you take English, or in some cases a foreign language? You might like, well, I'm a science major. Why do I need that? Ah, here's an example of that. So I'll also show you where history, we've kind of mentioned that some, history and English, we know math and the other sciences fit in, but things outside of the math and sciences are also very useful. Okay, so chromoplasts are more, you have them in more complex plants. Um, these chromoplasts don't have chlorophyll. And because they don't have chlorophyll, they're not green. They have other pigments in them. And a lot of times they're not used for photosynthesis like a chloroplast would be. They're used for attraction. Can you think of a part of the plant that is not green that is used for attraction? Petals. Okay, so Elena says petals. So petals contain chromoplasts. There's at least one other major thing um, that has this as well. Um, the inflorescence is made of flowers, so we should see petals. Yeah, that's kind of together, Elena. Um, it all depends. Sometimes the inflorescence parts itself that are not flowers may be colored. It's okay. Sometimes that stem that holds the flowers in the inflorescence might be colored. A lot of times it's green, depending. But I'm thinking about something else major. Okay, another part of a plant. Now, a lot of times you're probably not going to think, oh, that's a part of a plant, but you know it is. Okay, and I'm going to give you hints. You might find it in a grocery store. Actually, you will find some of these in a grocery store. And sometimes you get attracted to go eat them. Fruit, exactly. So think about an apple. The apple, the red part of the apple, um, they're not always red. Sometimes they're yellow, sometimes they're green. We think about various fruits that change colors. A lot of times they change from green to some other color, bright color. So one thing to note is that chloroplasts can actually degrade the chlorophyll and become chromoplasts. They can change. Okay, so we're going to hopefully look at a tomato and we'll be able to see some of the little red chromoplasts in the skin of that tomato. Okay. Leucoplasts fall under a couple of other things. We've got amyloplasts, and we'll look at amyloplasts in lab next week. These actually synthesize starch. Can you think of some plant that you eat that has lots of starch in it? Potato. Yep. And that's the one we're going to look at. Um, so potatoes, you can actually like stain those little starch amyloplasts, and you can actually see them under the microscope. We're going to do that next week as well. Elioplasts, we're not going to look at those, but they synthesize oils. They don't have any color. They're not pigmented. Okay, so that's different than chromoplasts and chloroplasts. Um, so basically, plastids arise from the division of mature plastids. So sometimes we'll see these developing from other kinds of plastids like uh, chloroplasts, but then they do other things. So on the left here, this is an amyloplast. It kind of looks a little gritty. Kind of, if you were to eat this, it might be kind of gritty, just a little bit like potatoes are just a little. Especially, has anybody ever eaten a raw potato? Nobody? Do y'all consider yourselves country? Nobody's had it. Okay.
A raw potato is too hard.
It's not actually. You peel the skin off. The skin's where you don't want to eat. I used to have a neighbor when I was really, really young, and he had a garden beside my house because our house was on one side and his garden started on the other. And when I was a kid, he would grow potatoes in that field, and he'd be out there on his tractor. And as a little boy, tractors, I'm going to be excited, just go look at the tractor. And he would hand me little potatoes, and he'd peel it off with his pocket knife and just give me the raw potato, and lots of good memories for me. But if you ever eat a raw potato, it's opposed to cooking it, it's kind of crunchy, a little bit gritty, and that's those amyloplasts that you're tasting. So I do not suggest that you eat lots of raw potatoes. It is not good for you. They are slightly toxic. Um, just heads up on that. Even the cooked potatoes are slightly toxic, and all of us contain some of those toxins within us, even from eating French fries. Just heads up. So, so you've seen people eat it, Brandy, that eat raw potatoes? Yeah, I don't suggest doing a lot of it. It was always like those little ones he would peel and give to me. I loved them. Um, I don't know. Maybe that's why I am the way today I am today. But I'm glad somebody else has seen that. Like I said, though, I don't suggest doing that a lot. And do not eat green skins of potatoes. That's where the highest amount of toxin is at. So if you've got green potatoes, at the very least, peel those things, get all the green off before you cook them. Okay. On the right here, you can see these are oil bodies inside of this, and that's from the lyoplasts. I see those in plants. I'm trying to remember. I think might be wild ginger or bloodroot. The seeds have little oil packets on it as a enticement for ants to come and pick up the seed and take it as dispersal mechanism. Okay. Now I put mitochondria in plastids. They're not actually plastids, but they function a lot in the same way. So we're going to talk about them as a group. They release energy from cellular respiration. They have two membranes, also an indication of endosymbiosis, as well as having their own DNA. And a thing to point out is that neither chloroplasts or mitochondria are able to function on their own. Even though they have their own DNA, they don't have all the DNA necessarily to survive by themselves. We'll talk more about that in endosymbiosis coming up. They have that inward membrane inside that gives a whole lot of different like whirly folds. They're called cristae. And again, this is all about increasing surface area so that you can produce more cellular respiration. And again, they arise from other plastids. So mitochondria and the other plastids, they are reproducing themselves, I guess is how I'd want to put that. Okay, which is another indication that they were at one point able to survive on their own. They're not able to survive outside the cell now, but inside the cell, they reproduce themselves. So when you go through cell division, that the point that the cell divides is not the point that mitochondria or chloroplasts divide. They actually divide at a different point in time during one of the growth phases of the cell. And so mitochondria make more mitochondria, chloroplasts make more chloroplasts. So if I were to ask you to draw an animal or a plant cell, and you put one mitochondrion in that cell, would that be representative of an actual cell? No. Okay, there's going to be more than one. I actually don't know the number that maybe is estimated inside cells. Could I give y'all a little bit of homework for Friday? A couple of questions that I want you to bring. Tyler, did you say no? I think she did. She said no homework. I just have two questions, just real quick things. You can Google at some point and see what you can find. Make sure you find a reputable source. Um, just see if you can find an estimation of the number of mitochondria in a human cell. And notice I'm saying
Can you
human.
say it again?
So find an estimation of the number of mitochondria in a human cell. Something's asking me if I'm playing music, and I'm not playing music, so I don't know what's going on. Okay. The second question is, and I'm going to give you a third question just so that we can have a good comparison. Okay. And the reason, why do you think I'm asking the question of the size of the human cell versus the size of a maize or corn cell? Why would I need to know that? I'm going to pick on Brandy. So, Brandy, why do you think that we need to know that comparison of those two cell sizes? Are you typing an answer? Okay. I definitely want you to see the range of size. But in reference to things like mitochondria, I would like to know, do we have a similar amount of, call it, mitochondria in each of those cells? Do we have a similar amount of mitochondria in a human cell versus what's in a corn cell? Does that make sense? And if you're like, oh, we've got 100 in human and 100 in a corn cell, okay, it's equal, right? Well, what if your corn cell is really huge and your human cell's a little bit smaller? then really who's getting more cellular respiration out of the deal? It's going to be the humans, right? So that's what we want to do. We have to make sure that we give ourselves a baseline and a comparison in which to look into these things. Okay, let's keep going. So here you can see the cristae, the what do you call it? The folds that gives us lots of surface area to do cellular respiration. This is just a link that you can use if you want to get a, it's kind of a little primer on endosymbiosis. I'm not going to get into a great detail of that. I've been kind of talking about it as I go. But if you need to refresh yourself, you can use this link or some other thing about endosymbiosis. Okay, you do want to understand it. Remember, cyanobacteria. Does anybody know what kind of bacteria were absorbed to create mitochondria? Or at least ones we think that were absorbed. Anybody? Purple bacteria. Okay, there's a group called the purple bacteria, and they're of the right size. They do the same kinds of energy production, so they seem to match up with what we have absorbed. Okay, also in the cell there are microbodies. These are small spherical bodies with a single membrane. Okay, which just means that they're not part of the endosymbiosis idea. They're distributed throughout the cytoplasm, and they do a lot of different things with special enzymes. Peroxisomes are ones that we will see. We won't talk about it much in botany, but in plant physiology, we'll talk about them because they're an intermediary organelle in photorespiration, something that keeps plants alive in harsh conditions like when things are too hot or too dry. They actually kind of connect the mitochondria and the chloroplast together. Glyoxysomes aid in converting fat to carbohydrates so that they can be used in cellular respiration. Vacuoles are something that animals have, but plants do something different. So in a lot of mature cells, 90% of the volume of that cell is going to be taken up by something called the central vacuole, or the great vacuole. It'll have its own membranes called the tonoplasts. It's filled with what's called cell sap, and this maintains pressure in the cell. Basically, it helps to inflate the cell, if you want to think of it that way. So it's maintaining structure through osmoregulation. And it frequently contains things that are water soluble, like certain pigments. Chlorophyll is a pigment. Anthocyanins, xanthophylls, things like that are pigments. So these might affect the color of the plant. It can also be used to store metabolic waste. So how many of y'all have had poison ivy at some point? Nobody? Y'all don't get out much, do you? So if you spend some time in the woods or even just in your yard where poison ivy shows up, oh, so Elaine, you're not allergic, so you can touch it. That's good. Be careful. There is a point that you might develop the allergy if you come in contact too often. So do be careful with that. The irritant that causes poison ivy, I think, is stored in the vacuole. Um, Not so much are you just touching the plant, but it's when you touch the plant and you definitely damage the plant and it breaks open cells, you end up releasing these metabolic wastes. Basically, it's the idea, if something were trying to eat this plant, these vacuoles would get popped open and those waste products, which we think of them as waste products for the plant, but they also end up becoming secondary products that get used in the plant defense sometimes too. So poison ivy is something, kind of an example of that. Whoops, there we go, cytoskeleton. So the cytoskeleton is a network of microtubules and microfilaments. Microtubules control the addition of cellulose to the cell wall, and microfilaments play a major role in the contraction and movement of cells. If you're an animal cell, We don't, of course, do that in plant cells because they don't need to be moving. They're fairly stationary, not completely, but fairly stationary. So what plants do is they use them for something called cytoplasmic streaming. And let me see if I can get this pulled up to show you. Of course, we've got a commercial. I remember the days when
writing's not that easy, but Grammarly can help. This sentence is grammatically.
Okay. We don't actually need to hear sounds. I'm going to turn that off. I want to show you. So this is an Elodia plant, and I am going to bring in, hopefully Monday I'll have some Elodia for us to use. A lot of people use it in their aquaria. Um, has anybody seen anything that looks like this before in maybe somebody's aquarium? Yeah, okay. Um, some of this grows out at the lock and dam, so I'm going to go get some. It's not going to look this nice because it's going to be covered in algae, which can also be fun to look at. But you see these really thin leaves? They're about 2 cell layers thick, and we can see this under the light microscope. Do you see the movement of the chloroplasts? Isn't that cool? And so those chloroplasts are moving along the cytoskeleton. Really, really, really neat. I made my own video of this once in graduate school, but the video was like 5 minutes long and the file was so large. And at the time, the little thumb drives were not big enough to hold the file size. So I had to leave it in Ohio. Uh, but so much fun to watch this. It's such great videos. I can't promise that ours are always going to be quite so excited. But if at all possible, I'm going to try to um, collect it not long before lab so that it has been out, in the sun. If it's not in the sunlight, those mitochondria are not, sorry, those chloroplasts are not going to be all that, what's the word I'm looking for? Movable, excited, because you know how I talked about how dim light and bright light affect where the chloroplasts are located? If I kept this in the dark, you're not going to see them moving as much. So hopefully if I can catch them pretty, pretty fresh for us, I'm hoping we'll be able to see something under the microscope pretty nicely. So that's my goal for next Monday. I just make sure I can get it before lab. Let's get back to our last slide here. And so this is just a picture of all the cell of plants. You can use it as a way to test yourself or kind of remind yourself. You can kind of look at each one of the things and then talk your way through like what it does. Okay, so that's one thing I would suggest doing. Do y'all have any questions on plant cells? No. Everybody good? Okay. I have about 10 minutes. I'm going to move on to the next slide. Is that okay? Or that's the next PowerPoint. Do y'all need to get anything off this before I move? Okay, I'm going to turn this one off and go get another one. That's not what I want. That's really slow. Come on, there we go. Okay, so plants of the day. Does anybody know this white flowered plant? You don't have to know its name, but have you met it before? Anybody? It's a pretty weedy plant. It's in lots of places. If you pay attention, you'll see it along the roadside. Nobody? Okay, let's see. Okay, good, Kiviana, you've seen it. Have you ever heard the name Queen Anne's lace? Okay, so this one's sometimes called Queen Anne's lace. Um, how about Daucus carota? That's the scientific name. How many of y'all have heard of carrot? This is wild carrot, Daucus carota. You'll see that in the name. So here's a guy with a wild carrot. I don't know why he's holding it, but that's what I wanted to show you right here. This is a normal, you know, domestic carrot versus a wild one. It may not taste as good, but it is a carrot and it is edible. Um, you can eat carrot leaves. I'm not sure, but I think there's some maybe medicinal uses for the carrot flowers here, the Queen Anne's lace flowers. Now, here's the thing. I'm going to make a point here because this is botany and we need to talk about this. If you aren't sure of a certain plant, you should not consume it. In fact, there are probably some plants, if you're not sure about, you shouldn't even touch them. Okay. This comes from a group of plants in the, I think it's APACE, that there are many, many poisonous plants in this group. Okay. There are also many toxic plants in this group. There's one called hogweed that you can touch and it causes your skin to become sensitive to sunlight and you can get major UV burns because of the chemicals from the hogweed. So, and this is huge, and I mean huge plants, taller than I am, and it grows wild. It's not native here, but it's growing wild here now. You have to be careful of it. I used to have to not hogweed, but there's another one. There's one called hemlock. It's not the tree hemlock, but the one that, who was it that had to kill himself with hemlock? The Greek guy. Does anybody know what I'm talking about? Was it Socrates? He had to commit suicide, and they gave him that to drink, but it came from hemlock. Also, I think it's in this group. So as you can see, don't eat anything that you aren't absolutely certain you know what it is. Okay, just disclaimer there. Does everybody agree to that? Don't put plants in your mouth that you're not sure about. Okay. Okay. Just I know that sounds like duh, but people do dumb stuff sometimes. I was reading a book about a guy walking, or in this case, hiking the Appalachian Trail. Lots of people do this. Yeah, don't do mushrooms unless you know what you're doing. I don't do mushrooms. Don't. Mm-mm. But I was reading a book on the Appalachian Trail. This guy hiking it, called Hiking Through, I think was the name of the book. And he decided to, look here, I think it's at home. He, for some reason, decided that every flower, every new flower he came across, he would eat it. I was yelling. I mean, this is at one point I'm reading this book and I'm like, stupid, stupid idea. Don't do that. And at one point he ate rhododendron flowers. Rhododendrons are toxic. Okay. So just so you know, azaleas are rhododendrons. They're a rhododendron species. Don't eat them. And I've even been told that if you're up in the mountains and you know they have those huge rhododendron stands, and if you're there at the right time of the year, you can see beautiful flowers. But in the rest of the year, it's just green. But don't even use a rhododendron stick to like cook a hot dog on over a fire. That can make you sick. Okay, you just don't do it. Okay, knowing what you're dealing with is really important. So just give you all those little heads ups. Okay, so there was our plant of the day. Let's talk about cellular reproduction. This is still some of the review. Y'all should know most of this, if not all. Cell division is part of what we call the cell cycle, is cell reproduction. It's divided into two phases, interphase and mitosis. Interphase is when you have the time when the cells are not dividing. You spend most of the time, if you're a cell, in interphase. The G1 portion of interphase is when the cells are increasing in size. The S phase is when the DNA replication takes place. By the way, does anybody have an idea of what G stands for? If the cell is increasing in size, what does the G stand for?
Growth.
Yep, it's growth or growing phase. What do you think S stands for? This one may not be quite as clear. Your DNA replicates. What do you think S stands for? You'll know the word when you hear it, but it may not be a word that you use all the time. Okay, give up. Okay, synthesis. Okay, when you synthesize to make. In this case, we're making more DNA. So we use the S, synthesis. I say this to help you connect the name G1 and S to a word that makes sense about what's happening. These are not just they didn't pick just random letters. They chose those letters based on what's happening. So growth 1, cells increase in size. Synthesis phase, DNA replicates. Growth 2, this is when your mitochondria divide. And your chloroplasts and your microtubules are produced to be able to pull the DNA apart into the two separate cells. This is all interphase, okay? Cell is not actively dividing, it's getting ready for division. So mitosis then refers to the process of cellular division where you get two daughter cells. They get an equal amount of DNA and a relatively equal amount of everything else in the cell gets duplicated during interphase and then divided when mitosis is finished. So you should end up with each daughter cell, which is 2, and an exact copy of the parent cell. Okay. In plants, mitosis occurs in a part of the plant called the meristem. These meristems are at the tip, the growing tip of a stem. Okay, very important point. Imagine what's going to happen to the growth of a plant if you cut the tip off of the stem. What do you think is going to happen to the growth of that stem if you cut the tip off? Will it grow it back? Will it stop growing? What do you think? It's going to stop. Yep. Thank you. So I'll give you an example. How many of y'all grow palm trees in your homes? Anybody grow a palm tree? Why not? Why don't you have coconuts in your backyard? This is an easy question. It's too hot here to grow palm trees, right? Are y'all really doing this? Is it too hot to grow palm trees? Where do palm trees grow? In Siberia, right? And all the snow? No. Where do they grow? Okay, at the beach. Yep. So why don't we grow them here? The beach is warm. It's warm here. Our problem is winter, okay, when it gets too cold. Now you're like, how do we go from meristems and mitosis to hot and cold? Well, here's the thing. Palm trees have one meristem, okay? So you can grow a palm tree here in the summer and be fine, but you leave it out over winter, and that winter will freeze it could freeze, not always. Sometimes we don't have cold enough ones to freeze it, but if we have a cold enough winter like this last winter where we had like a week of deep freeze, um, then what's going to happen is that poor meristem that it only has one of for a palm tree, when that gets frozen, no more palm tree. Does that make sense? Now, other plants have more than one meristem. So if they get one cut off or frozen, whatever the case may be, they can activate other ones. But not every plant can do that. And just I use the palm tree example because it's extreme. It has a single meristem and it grows upwards. And if you kill it, that the plant will die eventually because it can't continue to grow. I'm going to stop there for today. It's right about time to finish. Actually, it is time to finish. Um, and that's it.